Molecular oncology. So to talk about cancer, we first have to talk about the normal cell cycle because cancer is uncontrolled cellular growth. So in a typical cell cycle, we have the G1 phase where the cell grows and does its normal activity. Then the cell starts to prepare for mitosis or cell division. It will enter into the S phase, and during the S phase, that's when synthesis or DNA replication happens. So cells are going to grow really large and split in half. And when they split in half, they have to have equal copies of genetic material in each one of the cells. So we replicate the entire genome so that when we split these cells in half, each half gets equal amounts of DNA. Then we have G2, and this is the final growth phase for the preparation of mitosis. The cell is going to grow a little bit larger so that when it does split in half, there's enough space in each one of the sister or daughter cells in order to have all of that genetic material inside. Then we have M, mitosis. And mitosis has the four main stages: prophase, metaphase, anaphase, telophase, where the chromosomes are basically going to become visible, cluster up, align in the center, be pulled apart to opposite ends of the cell, and eventually new nuclear envelopes are going to form around each separate pile of DNA, so that the DNA is divided equally amongst both of the cells. And this is replicated DNA, so each cell will end up with the same amount of DNA as what it was in the G1. Then you have cytokinesis, the separation of the cytoplasms of the two daughter cells. G1 to S, so there's a point here where the cells arrest in G1 prior to starting DNA synthesis. G2 to M, where the cells arrest again prior to starting mitosis. Cancer results when the cell division cycle proceeds from G1 to S or from G2 to M phase inappropriately. So it skips ahead when it shouldn't be. Only cells that can divide will actually be able to develop cancer. If it's a non-dividing cell, it won't be able to develop cancer. Erythrocytes don't go through mitosis. They don't have genetic material anymore. When an erythrocyte dies, it's done. So you typically don't have cancer developing from mature erythrocytes. Neurons are another example. You wouldn't have cancer developing from neurons. So how do we get brain cancer? Well, brain cancer arises from those supporting cells that surround the brain, like astrocytes, microglial cells, the dividing supporting glial cells that surround the brain and spinal cord, not the neurons themselves. Muscle cells, like skeletal muscle, cardiac muscle, you're not going to have cancer of the skeletal muscle or cancer of the cardiac myocytes because those cells don't divide. When we have non-dividing cells or cells that never divide, those are in phase G0. And we wouldn't then stimulate them to become dividing cells. So some cells just can never become cancerous, but any normally dividing cell has the potential to become cancerous. Now, the more we make particular cells divide, the higher likelihood that there's going to be an error when we replicate the genetic material. Those errors are known as mutations. And mutations, if in certain genes, can cause this dysregulated cell cycle. Some mutations just straight up kill the cell. So a mutation happens and it's lethal and the cell goes away. Other mutations they don't have any impact on the cell at all. But if a mutation occurs in particular genes or regions of the DNA, they can develop into cancer cells. So oncogenes, as proto-oncogenes, normally promote cell division or cell survival. Oncogene mutations are usually a gain of function and dominant. Tumor suppressors are genes that normally arrest cell division. Tumor suppressor gene mutations are usually a loss of function and are recessive. So we have growth factor, the receptor, a mitogen produces normal cell growth. Here we have a mutation that happened, there's no mitogen, cell arrest or apoptosis, the cell dies. Here, no mitogen plus an oncogene is mutated, or a tumor suppressor gene is mutated, and then we have uncontrolled cell division. Now, things that can cause an increase in cell division would be like obesity. If we have a lot of nutrients, we're going to increase our cell division just naturally. If we have an increase in friction, like on a skin cell, if we're constantly causing friction in that area, we're going to increase cell division to replace the damaged cells. If we have chronic inflammation, like if we have liver inflammation, we're going to constantly have to repair those cells, which can predispose us to getting liver cancer. If we have chronic inflammation in the lungs, such as that's caused by smoking cigarettes or smoking anything, that can increase the inflammatory responses, which are going to increase cell death and increase cell division and eventually lead to the development of cancer or greatly increase the likelihood of the development of cancer. So molecular detection of disease targets tissue-specific markers, antigens, or gene rearrangements, disease-specific markers, translocations, point mutations, polymorphisms, and tumor suppressor or oncogenes, and viruses, Epstein-Barr virus, hepatitis C virus, human T-cell leukemia virus, type 1, HTLV1. Methods: hybridization, blotting, standard polymerase chain reaction, electrophoresis, quantitative PCR, and sequencing. Gene mutations, oncogenes, tumor suppressor genes, chromosome structural abnormalities like translocations, deletions, or insertions, and chromosome number abnormalities, aneuploidy or polysomy. Somatic mutations are caused by environmental factors, sporadic occurrence, not clustered in families or populations. Someone smokes, it leads to the development of sporadic mutations in lung tissue. Genetic mutations are inherited as a cancer risk. They are clustered in families and particular populations and may also have an environmental component. Examples of somatic targets: the EGFR gene family. HER2/neu. The HER2/neu gene encodes one of a family of human epidermal growth factor receptors. This gene is frequently amplified in breast cancer cells, resulting in increased amounts of HER2 cell surface protein. HER2 expressing tumors are sensitive to Herceptin, a monoclonal antibody therapy. HER2 protein is detected by immunohistochemistry. HER2/neu gene amplification is detected by fluorescence in situ hybridization. The EGFR oncogene encodes another of the same family of epidermal growth factor receptors. This gene is mutated or amplified in several types of cancer cells. Tumors with activating mutations in EGFR are sensitive to tyrosine kinase inhibitors. EGFR protein is detected by immunohistochemistry. EGFR gene and chromosome abnormalities are detected by FISH. EGFR gene mutations are detected by single-stranded conformation polymorphism, SSCP, sequence-specific primer PCR, SSPCR, or direct sequencing. So here's a couple examples already where when we're looking for cancer, we can look for the protein, the mutated protein, or we can look for the gene itself, the mutated gene. Somatic targets in solid tumors, downstream signals. Sometimes our targets are downstream. So we have this original pathway here and we can detect some things that would be on the outside of the cell, but what we're actually looking for is deep inside of the cell down a target. The Kirsten rat sarcoma viral oncogene encodes a key component of cell signaling. Mutation and KRAS are the most common oncogene mutations in cancer. KRAS mutations are associated with tumor malignancy and may affect response to some therapies. KRAS gene mutations are detected by PCR, qPCR, or direct sequencing. BRAF, the BRAF or VRAF murine sarcoma viral oncogene homolog B1, BRAF, encodes a key component of cell signaling. Mutations in BRAF can stimulate the RAS-RAF pathway in the absence of KRAS mutations. BRAF may affect response to some therapies. Mutated BRAF protein is also a therapeutic target. BRAF gene mutations are detected in the same methods that are going to be repeated throughout here: PCR, qPCR, direct sequencing. Ewing sarcoma, EWS, synovial sarcoma translocation, chromosome 18, synovial sarcoma breakpoint 1 and 2. So that's extended out there. Paired box forkhead in rhabdomyosarcoma, ataxia, telangiectasia mutated gene, von Hippel-Lindau gene are other somatic mutations associated with solid tumors. VMIC, rearranged during transfection, are more examples. P53, the 53 kilodalton protein encoded by tumor protein TP53 is a transcription factor. TP53 is mutated in half of all types of cancer. Losses of the TP53 function is an indicator of poor prognosis in colon, lung, breast, and other cancers. Mutant P53 protein is detected by immunohistochemistry. The TP53 gene mutations are detected by a variety of methods, including immunohistochemistry and direct sequencing. Inherited mutations in tumor suppressor genes are recessive for the malignant phenotype. Mutations in tumor suppressor genes are dominant with respect to the increase of malignancy. Loss of heterozygosity exposes the recessive mutant allele in hemizygous state. This is explained by the two-hit hypotheses. It can be detected by short tandem repeat STR analysis. Loss of a linked heterozygous STR implicates a concurrent loss of one gene allele. Inherited breast cancer risk. BRCA1 and BRCA2 are tumor suppressor genes encoding proteins that participate in DNA repair. Inherited mutations in BRCA1 or 2 significantly increase the risk of breast cancer at an early age. Frequently occurring mutations are listed here. And they're detected by SSP, PCR, sequencing, and other methods. Most mutations are detected by direct sequencing of both genes. Lynch syndrome, formerly hereditary non-polyposis colorectal carcinoma. Inherited cancer accounts for about 5% of colon cancer. LS is associated with mutations in genes encoding components of the mismatch repair system. So just a note on mismatched repair system, because it's mentioned here and then it's explained a little bit later. The mismatch repair syndrome system is when you are making copies of a strand of DNA, you're supposed to be copying it and making complementary bases on the opposite strand. Sometimes there is a mistake made where the wrong base that's not complementary is added into a growing chain of DNA. Now, typically that would be corrected by the mismatch repair system. But if there are mutations in the genes that encode that system, the proteins that run that system, then your mismatch repair doesn't happen. And that's when you can have those accidents that happen in the DNA and they don't get repaired. And if that happens in an essential gene that's supposed to control the cell cycle, then that can develop into cancer. Microsatellites 1 to 2 base tandem repeats are sensitive to errors during DNA replication. These errors are normally corrected by the MMR system. So here is an example. We're supposed to have a T here and it's not present. So that forms a bump. And then we would have the mismatch repair system come in here and correct it. So here's the mismatch repair system. Here you would have the chunk of DNA with the issue in it chopped out, and then you would have a better chunk put back in to repair the damage. Lynch syndrome and microsatellite instability. 85 to 90% of Lynch syndrome colon tumors have microsatellite instability, or loss of the MMR. MSI is also a prognostic factor in endometrial cancer. Mutations in genes of the MMR system, or loss of function, are inferred by testing for MSI. MSI analysis determines gene function. Direct sequencing is used to detect the actual gene mutation. MSI is analyzed by assessing the stability of at least 5 different microsatellite loci, and it's recommended by the National Cancer Institute. MSI is detected by comparing PCR amplicons of the microsatellite loci. Unstable loci appear as extra products in tumor tissue compared to normal tissue. Liquid biopsy, so circulating tumor cells can be assessed for diagnostic and prognostic mutations. Specialized collection methods may be used to collect cells by cell surface markers or eliminate white blood cells. Amplification methods are used to detect mutations. Cell-free DNA, RNA, free nucleic acid, exomes, specialized isolation systems, bead column concentration, targeted, directed at specific mutations, untargeted, gene panels, or whole exomes assessed from all nucleic acid collected. Molecular detection of leukemia and lymphoma targets antibodies, gene rearrangements, translocations, point mutations, polymorphisms, viruses. Methods: hybridization, blotting, standard PCR, RT-PCR, electrophoresis, PCR with heteroduplex analysis, SSCP, real-time PCR with gene or patient-specific probes. Gene rearrangements are normal events that occur in lymphocytes. Antibody genes, immunoglobulin heavy chain genes, immunoglobulin light chain genes, and T-cell receptor genes rearrange. This is what allows our immune system to detect all sorts of different types of pathogens. We have gene rearrangements that happens all the time when we're creating these receptors. And the two main receptors are: for B cells, it's an antibody. For T cells, it's the T cell receptor. Rearrangement occurs independently in each cell. So this shows a diagram of that progression. And these genes are rearranged as depicted here. Gene rearrangements may be used to detect leukemias and lymphomas arising from cells that have rearranged their immunoglobulin or T cell receptor genes. Clonality: normal lymphocyte populations are polyclonal with respect to Ig and TCR genes. So all different types exist. A leukemia or a lymphoma is monoclonal, monotypic, with regard to IgG or TCR rearranged genes. So if we see a lot of the same, or just one of the same type that exists, we can start to assume that it's a leukemia or lymphoma. Monoclonal populations are detected by sharp bands unique to the tumor cell population. Translocations for leukemia. So translocations are diagnostic and can be used for monitoring of hematological tumors. Examples are listed below. You see the T, the translocation, and then the regions that were translocated. And here's some more listed here. And lymphoma and myeloma translocations are listed here. Detection of hematological tumors, so translocations and other abnormalities in chromosome structure and number are detected by FISH. Translocation detection using FISH breakaway probe. And we talked about these previously in this class, where if they appear as separate, then they have the translocation. PCR or qPCR may be used to quantify tumor load during patient monitoring. FISH is recommended for initial diagnosis. PCR is better for monitoring. PCR may not detect all translocation breakpoints. FISH and karyotyping assesses large DNA regions, including all breakpoints. Quantification by qPCR or TACMAN. For qPCR, use a standard curve of tumor cells diluted into normal cells. For RT-PCR, qPCR, use a standard curve of transcripts of known copy numbers diluted in normal RNA. Translocations used in diagnosis and monitoring of hematological tumors. So T1418 is a reciprocal translocation between long arms of chromosomes 14 and 18. It's found in 90% of follicular lymphoma cases and 20 to 30% of large cell lymphomas. With translocation, the B-cell leukemia and lymphoma BCL2 gene is moved from chromosome 18 to chromosome 14. BCL2 is dysregulated and overexpressed when moved to chromosome 14. PCR detection, the forward primer hybridizes to chromosome 18, the reverse hybridizes to chromosome 14. The translocation is indicated by the presence of a PCR or qPCR product. Another translocation, T922, a reciprocal translocation between the long arms of chromosomes 9 and 22, is found in chronic myelogenous leukemia and acute lymphoblastic leukemia. This translocation forms a chimeric region or gene between the breakpoint cluster region BCR gene on chromosome 22 and the Abelson leukemia virus ABL gene on chromosome 9. The translocated chromosome is the Philadelphia chromosome. The chimeric gene region produces an abnormal protein that drives the tumor cell phenotype. FMS-related tyrosine kinase, internal tandem duplications close to the transmembrane domain, point mutations affecting an aspartic acid residue in the kinase domain, nucleophosmin, and CAAT binding protein. Detection of gene mutations in hematological malignancies, myeloproliferative disorders. So the Janus kinase, or JAK2, gene codes for a kinase enzyme and activates a single transducer, an activator of transcription, STAT pathway. Dominant mutation at position 617 of the JAK2 protein, V617F. A high proportion of patients with polycythemia vera, essential thrombocytopenia, excuse me, thrombocythemia, or idiopathic myelofibrosis carry JAK2 mutations. 4 mutations in the exon are listed. Myeloproliferative leukemia mutations are found in thrombocythemia. For example, acute myelogenous leukemia are listed here. Cancer gene panel testing, next generation sequencing, MGS, that should say NGS, not MGS, disease-specific panels, whole exome sequencing, arrays, comparative genomic hybridization, and gene expression arrays. Multiple gene panels, hotspot, frequently occurring known variants in different tumor types, tumor-specific profiles and hematology panels, large gene panels, 500 to 17,000 genes, 1,700 genes, single nucleotide polymorphism, SNP detection by mass spectrometry. Complexities of interpretation. So tumor heterogeneity. Mutations have a hierarchy. Variant allele fractions. Variants of unknown significance. So if there's a mutation there, we don't know if that mutation actually does anything. New advances in immunotherapy activate innate immune responses against tumor cells. And then you can review the summary.